The overall goal of the following experiment is to examine handwriting in individuals at risk for psychosis. This is achieved by recruiting two groups of participants. The first group is made up of healthy controls, and the second group is made up of individuals at risk for psychosis. As a second step, participants use a pen-enabled tablet computer to provide handwriting data, which is analyzed by software. Next, statistical analysis compares the ultra-high-risk participants to healthy controls on a measure of pen movement disfluency, called average normalized jerk. Results are obtained that show individuals at risk for psychosis show greater pen movement disfluency, a proximal measure for spontaneous dyskinesia based on significantly larger values of average normalized jerk.
The main advantages of this technique over existing methods, such as instrumental strain gauges or observer-based reports, is that it makes it easier for us to train raters and reduces experimental bias. In addition, we are able to compare trials across multiple sites, which is something we haven't been able to do until now. Demonstrating this procedure is Derek Dean, a graduate student from my laboratory. Tina Gupta, our clinical coordinator, will enact the role of a participant.
The first task is to find study participants through email, internet, and traditional ads. Recruit adolescent and young adult subjects with an ultra-high risk for development of psychosis, and healthy control subjects. Exclude all participants based on a history of head injury,